Welcome back. In this video, we're going to learn about DNA replication. So let's get started. The first thing we're going to learn about is an enzyme known as DNA helicase. This is an enzyme that breaks down hydrogen bonds between the complementary bases of two strands of DNA. So what does that mean? Okay, so here are two strands of DNA. So normally they're twisted. So let's untwist them and lay them flat. Okay. And so this little yellow triangle is meant to represent this enzyme. It's called DNA helicase. DNA because it only works on DNA, and helicase, ace means it's an enzyme. Hela because it's working on the helix, the DNA helix, the twisted part, going to do the untwisting. So it's working right here on these hydrogen bonds. So basically, it's going to go right down here, right down the middle. Think of it as a zipper. It's going to unzip all those hydrogen bonds between the A's and the T's and the T's and the A's and the C's and the G's and the G's and the A's. It's going to unzip all of them and go right down the two complementary strands of DNA. And I want you to realize that not only does DNA helicase go in one direction, it also, another DNA helicase can go in the other direction as well and unzip in both directions. And what this does, it gives us what is called a replication fork. So as it works its way down, do you see how we have like this little fork? This open area of DNA. In this case, it's going to give us a place where we're going to have DNA replication. But this is the exact same process that has to happen in order for transcription to occur. We'd have to open up the DNA using a DNA helicase before we can go from DNA to RNA. All right. So once we have our replication fork, we are going to develop what are called replication bubbles. So they're exactly what they look like. So the strands are not going to completely separate into two different strands. What is going to happen is we are going to have bubbles where it's going to unzip, remember, in both directions. And as it's unzipping, we're actually going to be attaching new nucleotides. Next slide, I'm going to show you how that occurs. And so what's going to happen is these bubbles are going to get bigger and bigger. And what you will see is there's multiple bubbles in each DNA strand. And eventually you can see that they will meet up. So in order to attach new nucleotides, we need a second enzyme. You can tell because it has an ASE at the end of it. It's only going to work on DNA. You can tell that because the name of the enzyme starts with DNA, and it's called DNA polymerase, meaning it's going to be making a polymer. It's going to be adding a bunch of little things. And what DNA polymerase does is it adds the complementary nucleotides to an existing nucleic acid strand. So what does that mean? It means if you've got something there, it can add to it. But you're going to go like, Dr. Powell, when we created this bubble, when we created this replication fork, there was nothing there to add to, right? There's nothing there. And I'd go, exactly. So we had to put something there first. And what happened first is this yellow thing. And this yellow thing was called a primer. So a primer gets added. And now the DNA polymerase can attach nucleotides to the primer. Okay. And so the nucleotides are going to be complementary to the nucleotides on the pre-existing DNA strand. Okay. And so this is going to happen on both sides the parental, the original DNA strands. So by the time we are done, these replication bubbles have gotten bigger and bigger. And by the time we are done, we will end up with a new DNA strand, the full length of the original old or parent DNA strand. And this occurs for each of the two original DNA strands. On this one, they're showing the original DNA strands as being blue and the new strands as being red. Since half of the original DNA strand still exists, this process of DNA replication is known by the term semi-conservative replication. It's not just called DNA replication because we're not taking DNA and Shazam making brand new DNA. We're keeping half of the DNA and semi-conservatively replacing it. Okay, so I've got 5 pictures here. I left you a bunch of words for you to practice and a bunch of pictures. As you were talking yourself through it the first time, there's words to help you, but I want you to make sure, and I added more words than I think are on your slides. So double check. Make sure you're using all of these terms, okay? And then as you get better at it, get rid of the pictures and make sure you can teach this process to a sibling, to a parent, to your dog, to your stuffed animal, to a peer. Okay, to make sure you understand this process. And that's it for this one. And I will see you shortly when we come back and we talk about the process of translation. Thank you so much for all your hard work in this course.